master's student from Texas A&M Kingsville. Today I am here to present my research project on efficacy of oxytetracycline for hormone management in South Texas River Plateau. Hormone is also known as citrus greening disease. Citrus greening disease is one of the most destructive diseases of citrus, causing significant economic losses worldwide. In Florida, due to the citrus greening, over the past 5 years, there are almost 1 million losses annually. The citrus greening is caused by a bacterium called Candidus Liberibacter asiaticus. It is a phloem-limited, gram-negative bacterium. The Candidus Liberibacter has 3 different species called Asiaticus, Americanus, and Africanus. The species Asiaticus and Americanus are spread by an insect vector called Asian citrus and the species Africanus is spread by an insect vector called African citrus. The location of the pathogen inside the vascular system of the tree has left the growers with minimal options to effectively manage the HLP disease. Oxytetracycline is a bacteriostatic antibiotic used for HLP management through tree trunk injections in Florida. HLP symptoms include blotchy mottling of leaves, small-sized fruits, seed abortion, canopy and twig dieback, coffee beans, and fruit drop. Objectives: evaluation of efficacy of oxytetracycline trunk injection against HLP through estimation of Candidus Liberibacter asiaticus titer by qPCR on leaf and root samples, and monitoring tree health by scoring tree HLP severity, canopy color, and canopy density on a grade scale of 1 to 5, and measurement of fruit yield and juice quality, and to test the OTC residues in the fruit and fruit juices after 6 months after OTC injection. My research study is located in commercial orchards of Edinburgh, Texas. It has 2 different experimental locations, Site 1 and Site 2. Both the experimental sites have a rare grapefruit variety on rootstock sour. The Site 1 has trees planted in the year 2008. They are of 17 years old, and the Site 2 has trees planted in the year 2016. They are 9 years old. And the total number of trees selected for a study in the Site 1 are 94, and the total number of trees selected for a study in the Site 2 are 96. Treatments. We have 4 different treatments. First one is UNC is an untreated HLP negative control. It is not treated any oxytetracycline. And second one is a untreated positive control. It is untreated with any oxytetracycline, but it is HLP positive. And there are 2 different treatments, treatment 1 and treatment 2. Treatment 1, trees are HLP positive and they are injected with oxytetracycline of 0.58 grams per tree. And treatment 2, these are HLP positive trees, these are injected with 1.1 gram of OTC per tree. And the commercial product used for trunk injection is Rectified. It has an active ingredient of 95% of oxytetracycline hydrochloride, and devices used for the tree trunk injection are called Ganget tree injectors, and we used 5 Gangets per tree. Each Ganget has a capacity of 20 ml of the solution, so each tree received 100 ml of solution and 5 Gangets per tree. Outline of the experiment. Initially, we injected the trees in the year June 2023, and for the next year, May 2024, for the 2 consecutive years. And we collected the samples of leaf and fibrous roots before the treatment of before the treatment of OTC and at 3 different points of treatment after OTC. At 3 months after OTC treatment, at 6 months after OTC treatment, and after 9 months of the OTC treatment. We collected leaf samples and fibrous root samples, and later we processed the leaf and root samples, DNA extraction and qPCR. Along with this study, we also assess the tree health through various parameters like canopy color, canopy density, and HLP severity on a grade scale of 1 to 5. And we also check for any damage at the site of injection due to the tangents we used for trunk injection. During the harvest, we measured the commercial fruit sizes and fruit diameters. And later, we extracted the juice and analyzed for the juice quantity. And finally, we recorded the yield of